You know that apple you had this morning? It wasn't just a piece of fruit. It was the result of literally thousands of years of genetic history. But here's the thing, that history is incredibly fragile. So today, we're going to dive into the critical mission to save the genetic library of our food, a mission to conserve what's called crop germplasm, which is the very foundation of our entire global food supply.
But what's the big deal? What really happens if this library of life that underpins our whole agricultural system starts to shrink? Well, this isn't some hypothetical question. It's one that history has already answered for us, and the consequences, they were devastating.
Back in the 1840s, Ireland depended almost completely on just one variety of potato, the lumper. So when a new disease, late blight, tore through the country, there was no genetic diversity to fall back on. No other potato variety had any natural resistance. The result was a catastrophic famine. It's just a terrifying reminder of the danger of putting all of our agricultural eggs in one genetic basket.
All right, so here's our roadmap. First, we'll decode this library of life and figure out what we're actually trying to save. Then, we'll explore the two main strategies, conserving plants out in the wild versus banking them for the future. And to wrap it all up, we'll look at the ultimate backup plan, a global failsafe designed to protect our food heritage.
Okay, so what exactly is this genetic treasure that we need to protect? It's way more than just a pile of seeds. It's a living, breathing library of biological information.
The official term scientists use is germplasm. The best way to think about it is as the sum total of a species' genes, all that raw genetic material, whether it's inside a seed, a plant cutting, or a tiny piece of tissue. It's the stuff that carries hereditary information from one generation to the next. You could say it's the essential toolkit for any and all plant breeding.
This diagram does a great job of breaking down how these resources are categorized. You've got what are called plant genetic resources, or PGRs for food, so our modern crops. Then you have resources for non-food things like wood or medicine. And this is critical. You have the wild genetic resources. See how they all overlap. And notice that term, landrace? That's a traditional variety that farmers have developed over generations, and it's often the perfect bridge between a wild plant and a modern crop.
And this right here shows you exactly why that wild diversity is so incredibly important. Plant breeders, they'll take these wild relatives, which are just packed with useful genes for things like stress resistance, and they'll cross them with those land races, which bring stability and nutrition to the table. The whole goal is to combine the best of both worlds into a modern, improved variety that gives us high yields, great quality, and the toughness to face future challenges like climate change.
So the main takeaway here is that we're trying to save a really wide range of materials. We're talking about those wild relatives, the land races that farmers have been perfecting for centuries, modern high-yielding cultivars, and even what are called obsolete cultivars. Now those are just modern varieties that have been replaced by newer ones, but they might hold some really valuable forgotten genes. All of it, including the breeding stocks that scientists use, makes up this priceless genetic pool.
Okay, so we know what we need to save. But how do we do it? Well, the first strategy is to save it right where it grows, out in the wild.
So there are two fundamental approaches here. First, there's in situ conservation, which basically means on-site. This is all about protecting plants in their natural ecosystems. The huge benefit here is that they can keep evolving and adapting to changing conditions. The other approach is ex situ, or off-site. This is where we take samples out of their habitat to protect them in controlled environments, like gene banks. This is our insurance policy against any immediate threats.
And within that in situ idea, there are a few different methods. You have biological reserves, which are big protected areas where wild crop relatives are just left to do their thing. There's on-farm conservation, which relies on farmers who keep planting and saving seeds from traditional land races. Even small home gardens can be amazing little hotspots of diversity. And then there's a cool term, circus situm, which means around the site. Think of a farmer who preserves valuable lone fruit trees within their fields. That's it.
And this brings us to an absolutely critical point. So much of this on-the-ground conservation isn't happening in some official reserve. It's happening in the fields of smallholder farmers all around the world. These farmers aren't just holding onto old seeds. They're active innovators. They are constantly experimenting, selecting, and adapting crops to their local environments. They are the everyday guardians of our planet's agricultural biodiversity.
But what happens when those natural habitats are threatened by development, or conflict, or climate change? That's where the second strategy, ex situ conservation, comes into play. It's about creating a safety deposit box for nature's genetic wealth. And the scale of this effort, it's just mind-boggling. Globally, there are over 7.4 million accessions. An accession is just a distinct sample of seeds or tissues, and they're stored in more than 1,750 gene banks. Every single one of those samples is a potential solution to a problem we might not have even encountered yet.
For most crops, the process is pretty straightforward, but it's incredibly precise. After collection, seeds are carefully dried to get their moisture content way down. Then, they're sealed up in airtight packets and stored in deep freezers at about -18 degrees Celsius. In those conditions, they can stay viable for decades, maybe even centuries.
Okay, but not all plants like to play by those rules. Some, like avocados or cacao, have what are called recalcitrant seeds, which means they just don't survive the whole drying and freezing process. And think about it. A lot of important crops, like potatoes or bananas, are grown from cuttings, not seeds. So, what do we do about them?
This is where biotechnology comes to the rescue with some seriously cool stuff. Using lab techniques, scientists can grow whole plants from just a tiny piece of tissue. And for long-term storage, they turn to cryopreservation, basically flash freezing those tissues in liquid nitrogen at almost -200 degrees Celsius. At that temperature, all biological activity just stops. It's like pressing pause on life itself. It's an incredible way to protect the plants that can't be saved in a simple seed packet.
Okay, so we've got thousands of these individual gene banks all over the world. But what happens if one of them fails? Because of a natural disaster, a power outage, or even a war? That's exactly why the global community created the ultimate backup plan.
Deep inside a mountain in the Norwegian Arctic, buried under permafrost, is the Svalbard Global Seed Vault. This is the final failsafe. Gene banks from all over the planet send duplicate copies of their seed collections here for safekeeping. It doesn't do any research. Its one and only job is to store these backups securely, just in case the absolute worst happens.
And we know this system works because it's already been put to the test. In 2015, the incredibly important Icarta gene bank in Aleppo, Syria, which held unique drought and heat-tolerant varieties of things like wheat and barley, became inaccessible because of the Civil War. But years earlier, Icarta had sent duplicates to Svalbard. They made the first ever withdrawal from the vault, and they used those backup seeds to regenerate their entire collection and restart their crucial work in new locations. It was a perfect and honestly a very powerful demonstration of why this global system is so essential.
So why does any of this matter to you? Well, because this preserved germplasm is the raw material we absolutely need to breed crops that can handle a changing climate. It's our best defense against new pests and diseases. It's the bedrock of global food security, and it's a living inheritance that we are protecting for every generation to come.
So, from a farmer's field to a high-tech lab, all the way to a vault in the Arctic, the mission to save our food's genetic heritage is happening as we speak. These collections are so much more than just a backup of the past. They are the essential toolkit for building a resilient future. Which leaves us all with one final question to think about. As our world keeps changing, how will we use this living library to design the foods of tomorrow?